Good morning, friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a systematic order, so it is quite convenient for the study. Now, in the last class, we are discussing about the development of the musculoskeletal problem, musculoskeletal development. And today, so we will see development of the skull, how actually skull of the fetus will go into develop. Now we know that what do you mean by paraxial mesoderm. In the last class we have seen. So let us take the paraxial mesoderm now. Now as the time passes, the segmentation of the paraxial mesoderm will take place at the cervical, thoracic and the lumbar areas. These segments are called as somites. The segments which are present over the most superior area is called as occipital somatomeres. As the time passes, the somites are divided into ventromedial and dorsolateral areas and their differentiation starts. In the ventromedial area, the epithelial cells becomes loose and becomes mesenchyme and start migrating towards the notochords and the neural tube area. And this is called as sclerotome. That is, in the ventromedial area, the epithelial cells are become loose and become mesenchyme, and then they start migrating towards the notochords area and the neural tube, and this particular portion is called as a sclerotome. Now the dorsal lateral area is called as dermomyotome, as they further moves down in the ectoderm and forms the dermis. The leftover portion is called as myotome, which is eventually differentiate into muscles. Sclerotome from every segment as they move medially, and they forms the vertebral column in the medial or the central area, while occipital somatomere sclerotome at the top gets converted into mesenchyme and distribute for the formation of the skull. Just lateral to the neural tube, special cells which are derived from the neural crushed cells will migrate in the different way. It migrates to upwards and contribute in the formation of some portion of the skull. Now skull has got mainly two parts. One is CNS, another one is development of the face. The neurocranium, that is vault of the skull, is a part of the cranium which is housing the brain. The viscera cranium is a part of the skull which is developing the bones of the face. Okay, now look at neurocranium. Pituitary gland is under the cerebral hemisphere, is called as is present under the cerebral hemisphere, which is called as base of the skull. Rest of the part of the neurocranium is called as vault of the skin. Neural crest cells will proliferate and provide the mesenchyme for the development of the vault and base of the skull. Mesenchyme derived from occipital somatomeres will proliferate and also covers the vault of the skull. These two layers will make the covers of the base and vault of the skull, which are initially fibrous membrane, fibroblast, then later they convert into bone portion. This happens by the direct pathway and indirect pathway. Now, at some areas, the fibroblast will convert into osteoblast and the bones start developing directly in the mesenchyme and they fuse each other without intermediate cartilage formation. This is called a direct pathway. This is called as a intramembranous ossification. So such type of bones are called as intramembranous ossifications. In certain other areas, the mesenchyme first convert into cartilaginous bone or cartilaginous tissue, and eventually that converted into bone tissue. This is called as endochondral ossifications. Now you can understand the difference between intramembranous and endochondral ossifications. In the development of the skull, both type of the ossification takes place. I mean, if you look into the skull, you have got both type of ossifications. Part of the cranium which is developing from the neural crushed cells, the bones are directly formed in the intramembranous ossification, and this part of the cranium is called as membranous part of the neurocranium. Part of the skull in the base of the skull, which is present in the base of the skull, the mesenchyme first convert into cartilaginous tissue and then it can convert into bone, that is chondrial ossification, and this area is called as a chondrocranium. So we have got neurocranium and chondrocranium. The area where the pituitary gland is present, that is called as sella turcica. We know that. The most cephalic part of the natal cord will be ending at the sella turcica. The most cephalic, most part of the natal cord will be ending at sella turcica. From the right and interiorly, the mesenchyme is derived from the neural crest. From the sella turcica, left and anteriorly, it is derived from the paraaxial mesenchyme. This area is called as precaudal plate, while the posterior part is called as caudal area. In the membranous part, when there will be formation of the bones, and these bones are held together by the sutures. The area where the sutures join together is called as frontanel. So there are sutures like sagittal, coronal, lambdoid, and metaphytic or occipital sutures, etc. The sutures basically made up of fibrinous material, which will give the scope of expansion when the brain is developing. Three important uses of the suture is: this helps for the molding of the head during the birth, so that baby can pass through the narrow vaginal canal, and it also helps to observe the increased or decreased intracranial tension, and also it gives the space for developing brain by expanding. Anterior frontal layer also gives the idea of the bone development. After birth, 19 months, mostly it will go into close. Now this anterior frontal layer helps in the development of the bones of the the development of the bones of the face, like maxilla, zygomatic, and some portion of the temporal bones, etc. Its development takes place from the first pharyngeal pouch. We have discussed the pharyngeal pouch, so which has got two components, upper and lower. Upper is dorsal, lower is ventral. Upper is developed rapidly when compared to the lower one. The pharyngeal pouch is a special connective tissue condensation which is developing from the posterior to anterior side. Dorsal component leads to development of the maxilla, zygomatic, and portion of the temporal bones. But the mesenchyme which contribute the formation of these bones is derived from the migrated neural crush cells. So facial bones are derived from mesenchyme of the neural crush cells. Even also there is formation of malleus and the incus, which are also derived from these neural crush cells. In the ventral part of the first pharyngeal pouch, there is a special cartilage formation called as Meckel's cartilage. This will not convert into bones because the development of the facial bone is intramembranous. There is no cartilages. Okay, directly bones are formed. Around the Meckel's cartilage, there is a mesenchyme which undergo proliferation and produce the intramembranous bone formation of the mandible. So these things are very important. Later, this Meckel's cartilage will disappear and convert into a ligament. The second pharyngeal pouch produces the stapedius of the inner ear. While discussing about the pharyngeal pouches also, we have seen. Then the bones of the face are very small. The bones of the face are very small when compared to the bones of the cranium at birth. The reasons are simple. They don't have the paranasal sinus. Underdeveloped jaw will be there. When the teeth develop, the growth will take place, and underdevelopment of the cheek is also there. Therefore, bones of the face are comparatively small when compared to the bones of the skull. Now, from the next class onwards, we will see some of the very important clinical aspects related to this development of the skull. Thank you very much.